In this final video on macromolecules, I want to talk about nucleic acids. So nucleic acids are the fourth category of macromolecules or biological molecules, those important molecules that make up living organisms, including us. Nucleic acids are composed of carbon, hydrogen, oxygen, we've seen all those, nitrogen, and phosphorus. And they come in two types. DNA is deoxyribonucleic acid, so that's where our DNA come from. And then RNA is ribonucleic acid. DNA serves as a library of genetic information. It's basically a bunch of directions that tells the cell how to make specific proteins, because everything is about proteins. Remember, that macromolecule is what you're made of after we take all the water out, mostly your protein. And then RNA is needed to make those proteins. RNA transcribes or copies the DNA information and then translates it into the amino acids that make up the proteins in protein synthesis. So the monomer of a nucleic acid is a nucleotide. And a nucleotide comes in three parts, or has three parts. There is a sugar part, and so here we have an example, deoxyribose is the sugar shown here. There is a phosphate group, so you see the phosphorus in the center here, phosphorus with some oxygen around it making up phosphate. And then there is a nitrogenous base. And it does have nitrogen in it. We can see the nitrogen over here on the right. But it's not an amino acid or a protein. It's another structure with nitrogen. So we call it a base or a nitrogenous base, or sometimes you'll even see a nitrogen base. Nucleic acids form long strands with the sugar from one group connecting to the phosphate of the next, making what's known as a sugar-phosphate backbone. So you can see we have this little bond sort of sticking up, and that's where the sugar would connect from the next nucleotide if we were building a strand of either DNA or RNA, in this case DNA because we have deoxyribose. And down here, this on the oxygen, we see another sort of unclaimed bond waiting, and that's where that phosphate group would connect. And so one sugar would connect to one phosphate, which would connect to the next sugar, connect to the next phosphate. And so until you make a long strand. The nitrogenous bases come in 5 different types, and so they make 5 different nucleotides. And we use the letters C, G, A, and T along with U to distinguish those different nucleotides. These 5 bases can be further divided into 2 groups, the pyrimidines or the purines. And you can see in structure, excuse me, the pyrimidines have just a single ring structure where the purines have a double ring structure. So our C is cytosine, T is thymine, U is uracil, and this is only found in RNA, so this one is kind of the oddball out. It replaces thymine. Thymine is only found in DNA. The other 3 are found in both RNA and DNA. Out of our purines, we have guanine and adenine. So DNA, I'm sure you remember from biology, forms a double helix, sort of a twisted ladder shape. And it does that double helix because we have hydrogen bonds holding base pairs together. So the bases will pair up, will connect in a certain order. A's will bond with T's and T's with A's, and C's with G's or G's with C. So adenine with thymine, cytosine with guanine. These base pairs then form the rungs on the DNA ladder. So we see our base pairs here forming a rung, and the dotted line in the middle is representing the hydrogen bond. A hydrogen bond is an attraction between hydrogen and a large molecule such as oxygen or nitrogen, and it's enough to hold things together, but it's not as tight as the covalent bond that actually makes up these individual molecules along the strand. So DNA stays zipped together. The hydrogen bond holds it together pretty well, but it also can be unzipped so that we can read the genetic code on either strand. RNA forms only a single strand, but it can match up to DNA when translating a gene to make a protein. So the A on DNA will bond with a U on RNA, and all the other bonds are the same. The T on DNA bonds with an A on RNA, and C's and bond with G's. There are actually 3 types of RNA that are needed for protein synthesis, and the shape, they come in different shapes. and those shapes are formed by the attractions between the different nitrogenous bases, just like we see the different shapes of proteins being formed by attractions between the atoms in those molecules. So here's a large diagram that's just providing an outline of protein synthesis. That we have start off with our DNA in the nucleus, and it opens up and a molecule of messenger RNA is made that translates the coding that's on that DNA strand, the order of the nucleotide bases. That mRNA then leaves the nucleus and it heads out into the cytoplasm where it runs into a ribosome. So here we have a ribosome. It comes in two parts, and those two parts clamp down around this mRNA. And ribosomes also are made of RNA. So we have rRNA for ribosomal RNA, mRNA for messenger RNA. And while the messenger RNA is in the ribosome down here in the bottom, we see these sort of T-shaped structures, and these are known as tRNA or transfer RNA, and they carry in the amino acids that match up with the codes that's on the mRNA. So here we have one that we see is carrying a particular amino acid, and in the ribosome then those amino acids are joined together in a peptide bond to make a long peptide chain that will eventually fold into a protein. There are other nucleotides that are not part of nucleic acids that are also very important in metabolism and keeping living organisms alive. They are involved in cellular respiration, which releases energy from the chemical bonds in food molecules. They're involved in cell signaling, which is basically starting and stopping the various chemical reactions that keep everything alive. And other nucleotides are enzyme cofactors, which basically means they serve as helpers to enzymes, helping, enzymes help chemical reactions happen faster, and some enzymes actually need other molecules to help them get started, and those are known as cofactors. So some examples of these type of nucleotides, and they often are, because their names are quite long, we use just the letters to abbreviate what the words are. So we have CAMP, which is a signaling molecule. It's one of the ones that gets a chemical reaction started inside a cell. NADP is an enzyme cofactor, so it's going to work with an enzyme to help some chemical reaction happen. And then ATP, which we heard about a lot in biology, which is our energy currency of the cell. It's how we pay for things with energy in the cell. So just to end with a brief review of ATP, I like to describe it as a rechargeable battery for cell activities. It can take a tiny little bit of energy, just the right amount that's needed for the various things that have to happen inside the cell. The ATP molecule has 3 phosphate groups, so not just one like most nucleotides, but 3, and that allows it to store a lot of chemical energy in those bonds between the phosphate groups. When those bonds are broken, an ATP actually becomes ADP, which is adenosine, instead of adenosine triphosphate, tri meaning 3, it becomes adenosine diphosphate, having only 2. But during that process, a lot of energy is given off, cell-wise speaking, and that energy then powers something that needs to happen. The ATP then is recharged through the process of cellular respiration. The whole point of cellular respiration is to put energy into ATP molecules. And every human cell is estimated to use at least 1 billion, possibly as many as 2 billion ATP molecules a minute. So through the course of the day, you use and reuse, because it's recyclable, about your body weight in ATP a day. It's a very important molecule. If you didn't have ATP, you're not going to remain alive. So that concludes my brief overview of nucleic acids and this one important nucleotide that's not part of nucleic acids, but certainly is part of keeping things alive. This finishes up the discussion of the macromolecules.